I want you to picture a metropolis, but not the standard static image of a city skyline. Picture a bustling, infinitely complex metropolis, humming with just an unimaginable density of traffic, energy, and, well, communication.
Right.
In this city, you have high-speed trains hurtling along perfectly insulated subterranean tracks. Those are the electrical signals of your brain. And then you have the bustling transfer stations, these massive, chaotic, highly regulated hubs where the currency of information physically changes hands, is translated into a completely different language, and is sent onwards to 1,000 different destinations.
Which would be the chemical synapses.
Exactly. For a long time, the public understanding of the brain has relied on the metaphor of a static computer, just wires, hard drives, and circuit boards. But when you really look closely at the biology, it is so much more dynamic, so much more fragile, and so much more alive than a piece of silicon.
Yeah, absolutely.
Welcome to the Deep Dive. Today, we're taking a microscopic lens to Kaplan and Sadok's comprehensive textbook of psychiatry. Our mission here, to figure out how the physical microscopic chemistry in your brain somehow manages to create your mind.
It is the fundamental question of neuroscience, isn't it? I mean, the reason we are exploring the cellular and synaptic basis of neural signaling today is because, quite frankly, you cannot practice modern psychiatry or neurology without it.
Right.
As clinicians or scientists or just anyone motivated to understand mental health, we have to grasp how this metropolis actually operates at ground level. We are moving beyond macro-level psychology today.
Because usually when we talk about psychiatric symptoms like major depression, crippling anxiety, a psychotic break, we talk entirely in the realm of subjective experience. We talk about how someone feels.
Exactly. But to truly understand how an antidepressant works, how psychotherapy physically remodels the brain, or how brain stimulation therapies like ECT actually function, you have to understand the cellular language they are speaking. We have to explore the neuron as both an electrical and a chemical entity.
So today we're looking at those exact same symptoms as tangible traffic jams, or misdirected trains, or structural collapses within this microscopic network.
Spot on. We are connecting the mechanism to the manifestation and finally to the clinical meaning. And to do that, a core conceptual framework we need to establish right away is the difference between intracellular communication and intrinsic excitability.
Okay, let me make sure I have the basic definitions down. Intracellular communication is how neurons talk to each other, like sending a chemical text message across the physical gap between two cells.
Precisely. That is synaptic communication, which gets the vast majority of the attention in popular science. But equally important and historically somewhat overlooked is intrinsic excitability.
Meaning the cell itself.
Right. This is about how prone a single individual neuron is to firing in the first place, completely regardless of what its neighbors are shouting at it. It is the internal threshold, the cell's own resting state.
Wow.
We are learning that psychiatric disorders frequently stem from dysfunctions in these precise signaling pathways. It's not just the chatter between the cells that goes wrong, it's the internal baseline calibration of the cells themselves.
That makes total sense. Because before a train can even leave the station, before any signal is sent, there has to be a power source. You can't have a high-speed rail network without a massively charged electrical grid just waiting to be unleashed.
No, you can't.
Which brings us to the resting state of the neuron.
Right, the resting membrane potential. This is the potential energy available for neuronal signaling. And to understand it, you have to understand gradients.
Like chemical and electrical gradients.
Exactly. Imagine the cell membrane not just as a wall, but as a highly selective, heavily guarded barrier with very specific doors. The key players trying to get through those doors are ions. So, charged particles, specifically sodium, potassium, calcium, and chloride.
And when the cell is just sitting there, completely at rest, not sending any signals, the source material says the most important door that's left propped open is the potassium door.
Correct. The cell membrane is studded with what we call non-gated or leak potassium channels. They are essentially always open.
Always letting things through.
Right. Now, inside the biological environment of the cell, you have a very high concentration of potassium ions. Outside the cell, in the extracellular fluid, the concentration of potassium is quite low.
Oh.
So just based on pure physics and diffusion, potassium naturally wants to leave the cell. It wants to flow down its chemical gradient from an area of high concentration to an area of low concentration.
Like people pouring out of a packed stadium after a concert just trying to find some empty space.
That's a great way to look at it. But here is the critical twist. Potassium ions carry a positive electrical charge. And inside the cell, you also have a massive amount of large, negatively charged proteins, amino acids, and nucleic acids.
And they can't get out.
No, these molecules are huge. They cannot fit through the doors. They are essentially trapped inside.
So as the positively charged potassium ions flee the crowded cell, they are leaving all those massive negative proteins behind.
Yes. And because positive charge is leaving, the inside of the cell becomes increasingly negative relative to the outside. This creates our second force, an electrical gradient.
Ah, because opposites attract.
Exactly. Because opposite charges attract, that growing negative environment inside the cell starts acting like a magnet, trying to pull the positively charged potassium ions back inside.
So the single potassium ion is caught in a brutal tug of war. The chemical gradient is screaming, it's too crowded in here, get out. And the electrical gradient is screaming, but we're negative and you were positive, come back.
It is a perfect, continuous biological tug of war. And there is a precise mathematical point where those two opposing forces perfectly balance out, where the chemical push exactly equals the electrical pull, resulting in zero net movement of potassium.
And there's an equation for this, right?
Yes, this is calculated using the Nernst equation, which gives us the equilibrium potential for any given ion. For potassium, under normal physiological conditions, that equilibrium potential is around minus 90 millivolts.
Minus 90.
If the cell membrane were only permeable to potassium and absolutely nothing else, the resting potential of your neurons would be exactly minus 90 millivolts.
But biological systems are never that clean and simple, are they? I mean, the membrane isn't perfectly sealed.
No, it isn't. And that's where the textbook introduces the Goldman-Hodgkin-Katz, or GHK, equation. The GHK equation calculates the actual resting potential of the neuron because it factors in the reality that the membrane is slightly leaky to other ions as well, particularly sodium and calcium.
And sodium and calcium are mostly hanging out outside the cell looking to get in.
Right, and they are both positively charged. Furthermore, because the inside of the cell has become deeply negative thanks to the escaping potassium, both the chemical gradient, because they want to move to an area of low concentration, and the electrical gradient, because they are drawn to the negative interior, are screaming at sodium and calcium to rush inside the cell.
Even through tiny leaks.
Even though their specific leak channels are mostly closed at rest, a tiny bit of sodium inevitably sneaks through the barrier.
And because sodium is positive, that tiny leak into the cell pulls the overall voltage away from the extreme minus 90 millivolts, right? It makes it a little bit more positive.
Exactly. That steady, tiny influx of positive sodium pulls the actual resting potential of a typical neuron up to somewhere between minus 55 and minus 70 millivolts.
Okay, but hang on. This seems like a fatal design flaw. If the cell is sitting there at minus 70 millivolts, but it's constantly leaking, potassium is always trickling out, and sodium is always trickling in. Over a few hours or even minutes, wouldn't those gradients just completely equalize? The battery would just bleed out and die?
It absolutely would. If the neuron relied purely on a passive diffusion, the gradients would run down, the voltage would drop to zero, and the cell would die.
So how does it survive?
To prevent this catastrophic failure, the neuron employs a massive, relentless piece of molecular machinery, the sodium-potassium ATPase, commonly just called the sodium pump.
Let me try an analogy here. If the resting cell is like a wooden boat on the ocean, and sodium is the seawater constantly leaking in through the floorboards, while potassium is, fresh water leaking out into the ocean.
Sure.
The sodium-potassium pump is like a crew of frantic city maintenance workers in the boat, constantly bailing the seawater out and hauling the fresh water back in just to keep the boat afloat and perfectly balanced.
That is a highly accurate way to picture it. Mechanistically, for every single cycle this pump completes, it physically binds 3 sodium ions from inside the cell and forces them out into the extracellular space.
Okay, 3 out.
Simultaneously, it grabs 2 potassium ions from the outside and drags them back in. It is actively shoving these ions against their natural gradients. And notice the maths. It moves 3 positive charges out, but only brings 2 positive charges in.
So it's constantly losing a net positive charge.
Yes, we call it an electrogenic pump. By constantly expelling more positive charge than it brings in, the pump itself actively contributes to maintaining that negative resting potential of minus 70 millivolts.
But wait, if this pump is constantly bailing water against the tide just to maintain the baseline status quo so the neuron can simply exist at rest, how does the cell afford that? That sounds exhausting.
This is where the sheer biological reality of the brain becomes staggering. To run this pump, the cell has to hydrolyze ATP, which is the fundamental energy currency of all biological life. According to the textbook data, up to 40% of the brain's entire oxygen consumption is dedicated solely to running this sodium-potassium pump.
Wait, 40%. Almost half of the oxygen I am breathing in right now, sitting here talking to you, is being used just to run microscopic water balers, so my neurons don't spontaneously discharge.
40%, just to maintain the resting gradients. Just to keep the tension on the bowstring, so to speak, so the neuron is ready to fire when a signal finally arrives.
That is unbelievable.
And if we connect this physiological mechanism to clinical medicine, it explains a tremendous amount. It explains exactly why the human brain is so uniquely and terrifyingly vulnerable to hypoxia.
Because if someone stops breathing or has a stroke, you cut off the oxygen.
You cut off the oxygen, which means the mitochondria instantly stop producing ATP. Without ATP, the sodium-potassium pumps freeze.
Oh, wow.
Within seconds, the sodium leaks in, the potassium leaks out, the gradients completely collapse, and the neurons depolarize uncontrollably. They dump toxic amounts of neurotransmitters, calcium floods the cells, and irreversible brain damage begins within minutes. The massive metabolic requirement of this single pump links the microscopic cellular signaling we're discussing directly to whole brain survival.
Do we have medications that specifically target this pump? Because it seems like if you mess with it, you mess with the fundamental foundation of the nervous system.
We do, but interestingly, you see it used most prominently in cardiology, which highlights how these cellular mechanisms are conserved across different organ systems. Cardiac glycosides, like the drug digoxin, actually inhibit this exact sodium-potassium pump in the heart muscle cells.
Why on earth would you want to stop the pump in the heart?
It's a brilliant indirect mechanism. By partially blocking the pump, you allow a little bit of extra sodium to build up inside the cardiac cell. Now, the cell has another mechanism, a sodium-calcium exchanger, that uses the sodium gradient to push calcium out.
Okay, I think I see where this is going.
If the sodium gradient is weakened by digoxin, that secondary exchanger slows down. The results. Calcium builds up inside the cardiac muscle cell. And as we will discuss later, intracellular calcium is the trigger for muscle contraction. So by tweaking the resting sodium pump, digoxin increases intracellular calcium, which forces the failing heart to pump with much stronger, more forceful contractions. It is a masterclass in exploiting cellular gradients for clinical benefit.
That is fascinating.
Yeah.
Okay, so we've established the resting state, we've built the battery, the metropolis has power, but potential energy, a tightly pulled bowstring, isn't in action, it's just waiting. How does the neuron actually use this tension, this minus 70 millivolt baseline, to send a signal?
We transition now from the static energy of the resting state to the explosive reality of action. We need to talk about the action potential.
The spark, the train finally leaving the station.
Exactly. Let's walk through the exact biophysical mechanism. The resting membrane is governed by those leak channels, but the membrane is also packed with voltage-gated channels. These are doors that are bolted shut at minus 70 millivolts, but they are highly sensitive to electrical changes. If the neuron receives an excitatory stimulus from a neighboring cell that causes the inside of the cell to become just a little bit more positive, say depolarizing from minus 70 up to about minus 45 or minus 30 millivolts, it hits what we call the threshold potential.
And what physically happens to the protein door at threshold?
At threshold, the structural conformation of the voltage-gated sodium channels snaps open. Because sodium is highly concentrated outside and the inside is still relatively negative, the electrical and chemical gradients finally get their way.
The floodgates open.
Yes. Sodium rushes into the cell in a massive, rapid wave. The sudden influx of positive charge causes a spike in voltage, a rapid depolarization. The membrane voltage shoots up past zero, reaching positive territory, usually peaking around plus 40 millivolts. This peak is called the overshoot.
But the cell can't stay at a plus 40 millivolts. That would be chaos.
Yeah.
The signal has to pass, the cell has to reset.
Right. And the cell has two distinct immediate mechanisms to bring the voltage crashing back down to reality. First, those voltage-gated sodium channels don't just stay open until the voltage drops, they rapidly inactivate.
How does that work?
They possess a physical molecular plug, a chain of amino acids on the inside of the cell that swings up and physically blocks the pore just a millisecond after it opens, stopping the sodium influx dead in its tracks.
Even though the cell is still technically at a positive voltage.
Yes, the inactivation is completely independent of the voltage returning to normal. It is purely a time-delayed mechanical plug. Second, a different set of voltage-gated channels finally opens up, the delayed rectifier potassium channels.
They are called delayed because they are structurally a fraction of a millisecond slower to react to the initial threshold voltage than the speedy sodium channels.
Precisely. They finally swing open right at the peak of the action potential. And now, think about the gradients. Potassium naturally wants to leave the cell anyway because of the concentration gradient. But now, because the inside of the cell is at plus 40 millivolts, the electrical gradient also wants to violently eject the positively charged potassium.
So they both want it out.
Yes, so potassium rushes out of the cell in a massive wave. This exit of positive charge rapidly repolarizes the cell, dragging the voltage down from plus 40 past 0 and heading back towards minus 70.
In fact, reading the text, it seems like the delayed rectifiers overcompensate. They pull the voltage down so hard it crashes right past the minus 70 baseline into an undershoot.
Yes, the after hyperpolarization. These potassium channels are a bit sluggish to close once the voltage drops. So for a brief moment, the cell continues losing potassium and becomes even more negative than its usual resting state, drifting down towards that theoretical minus 90 millivolt equilibrium potential we calculated earlier.
And this rapid sequence of events, the spike, the crash, the undershoot, creates a biological necessity called refractory periods, which, if I understand correctly, is essentially the neuron needing a moment to catch its breath.
It is more than just catching its breath. It is a critical safety feature to dictate the direction of the signal. Right after the peak of the action potential, the neuron enters the absolute refractory period. During this window, no matter how aggressively you stimulate that neuron, it is physically impossible for it to fire another action potential.
Why? Is it out of energy?
No, it is a structural limitation. Remember that mechanical plug that swung shut into the sodium channel pore? The channel is in an inactivated state. It cannot be forced open by voltage. It must wait for the membrane to fully repolarize so the plug can swing back out, resetting the channel to a closed but ready state.
And then there's the relative refractory period.
Yes, which corresponds to that undershoot phase, the after hyperpolarization. The sodium channels have reset so the neuron can technically fire again. But because the slow-to-close potassium channels are still leaking positive charge out, keeping the cell deeply negative, you need a massively stronger excitatory stimulus than normal to drag the voltage all the way back up to threshold.
I see.
This ensures that action potentials are discrete, separate signals, not just one long blended blur. And crucially, it prevents the electrical signal from traveling backwards up the axon toward the cell body.
Okay, so we've generated this intense, rapid spike of voltage, the action potential. Usually this happens right near the cell body at a specialized area called the axon hillock. But the signal has to travel down the length of the axon to reach the terminal.
That's right.
And in a human body, that axon could be incredibly long, like a motor neuron traveling all the way from your lower spine down to your big toe. How does the electrical train actually travel down the tracks without losing momentum?
To understand this, we have to look at how different types of neuronal fibers conduct electricity. If you look at dendrites, the branch-like structures that receive signals conduction is passive and decremental. It's like dropping a pebble into a pond.
Meaning it fades.
The electrical ripple is strongest at the point of impact, but as it spreads outwards, the current leaks out of the uninsulated membrane and the signal fades over distance.
That works for tiny distances within a cell body. But for an axon sending a signal down leg, passive fading won't work. The signal would die before it reached the knee.
Exactly. Axons need to transmit the action potential with high fidelity over long distances. Historically, in simpler organisms, or in our specific pain-sensing fibers, we use unmyelinated conduction.
Think of an unmyelinated axon like walking down a long suburban street, and you have the job of knocking on every single door to deliver a message. It is slow, tedious work. Every single microscopic segment of the axon membrane has to undergo that entire process we just talked about. Opening sodium channels, depolarizing, opening potassium channels, repolarizing. It's a continuous burning fuse.
That's a perfect visualization. And because it requires opening millions of individual channels in a continuous wave, it is incredibly slow. Unmyelinated fibers transmit signals at roughly 0.3 meters per second. That is a sluggish walking pace.
Yeah, that's not going to win any races.
If your entire brain relied on unmyelinated fibers, complex thought and rapid reflexes would be impossible.
Which is why evolution invented the high-speed subway tunnel, myelin.
Yes. Myelinated axons are wrapped in thick, tightly coiled sheaths of fat provided by support cells, oligodendrocytes in the brain, and Schwann cells in the body. Myelin is a brilliant electrical insulator. It completely prevents the electrical current from leaking out of the axon into the extracellular fluid.
So if the current can't leak out, it just rockets straight down the pipe. But the textbook says it's not quite a continuous tunnel.
No, it's not. The myelin sheath is interrupted at regular intervals by tiny microscopic bare patches called the nodes of Ranvier. And here is the genius of the system. The voltage-gated sodium channels aren't spread out evenly along the axon. They are densely, exclusively clustered only at these bare nodes.
So returning to the analogy, if unmyelinated conduction is walking down the street knocking on every door, saltatory conduction in a myelinated axon is like taking the express subway train.
Exactly.
You bypass all the local stops. The electrical current flows passively, effortlessly, and almost instantly through the heavily insulated myelin segments. When the signal starts to fade just a tiny bit, it hits a node of Ranvier, a major transit hub. The sodium channels there burst open, a fresh action potential fires, and the signal is instantly regenerated, completely refreshed, before rocketing down the next insulated subway tunnel.
The term saltatory actually comes from the Latin saltare, meaning to jump or dance. The signal appears to leap instantaneously from node to node, and the difference in biological speed is breathtaking.
How fast are we talking?
Well, while the unmyelinated fiber chugs along at 0.3 meters per second, a thick myelinated axon can transmit an action potential at up to 100 meters per second. That is hundreds of times faster.
So if we pull back to clinical psychiatry and neurology, why are we spending so much time talking about the physics of fat insulation on a wire?
Because it paints a stark, terrifying picture of what happens in demyelinating diseases. Think of multiple sclerosis in the central nervous system, or Guillain-Barré syndrome in the peripheral nervous system. In these disorders, the patient's own immune system mistakenly identifies myelin as a foreign invader and attacks it, literally stripping the insulation off the wires.
And if you strip the insulation, the subway tunnel collapses.
Exactly. Suddenly, the electrical current traveling down the axon leaks out into the surrounding tissue. The signal fades. And because there are no sodium channels in the newly exposed areas to regenerate the signal, the action potential simply dies before it can reach the next node. The train derails. But it is not just a straightforward loss of speed or strength. The true devastation is that it destroys the high-fidelity timing of neural networks. The brain is an orchestra. It relies on split-second, microsecond synchronization across vast distances.
Right, the timing is everything.
If a visual signal arrives at the processing center a few milliseconds late, or if a motor command is delayed, the entire network degrades. This loss of precise synchrony leads to the profound sensory, motor, and eventually cognitive deficits we see progressing in MS patients.
Okay, that makes complete sense. We have the resting battery, and we have the high-speed action potential rocketing down the insulated wire. But if I push back on this for a second, an action potential is essentially binary, right? It's a gunshot, it's an all-or-nothing, on-or-off biological switch.
It is entirely binary.
If the basic unit of communication is just a single, unvarying binary switch, how on earth do we get the staggering nuance of human thought? How do we differentiate between the sharp pain of a burn, the subtle memory of a childhood smell, or the complex emotional architecture of a manic episode? It can't just be one type of generic door opening and closing. The city needs more than one type of gate.
You've hit on the exact reason why we must explore Section 3 of our deep dive, the gatekeepers. The vast, almost intimidating diversity of ion channels. There are roughly 150 different voltage-gated ion channels encoded in the human genome.
150.
To give you a sense of scale, the only protein families that are larger are G-protein-coupled receptors and protein kinases. The sheer variety of these channels is what allows the brain to perform incredibly complex, nuanced computations rather than just acting as a simple light switch.
Let's start with the sodium channels, the instigators, the ones that cause that explosive upstroke we just talked about. The textbook goes into heavy structural detail here.
And the structural biology is vital for understanding how psychiatric drugs work. The main pore-forming unit of a sodium channel is a massive, complex protein called the alpha subunit. It consists of 4 repeating domains strung together. Think of a domain as a distinct block of the protein. Each of these 4 domains weaves in and out of the cell membrane 6 times.
Let me try to visualize this. Imagine the cell membrane is a thick, impenetrable stone wall. The protein is like a long piece of thread that a tailor has stitched through the wall, in and out, 6 different times per domain.
That is a perfect structural analogy. Now, the really clever, functionally vital part is the sequence of amino acids located exactly between the 5th and 6th stitch. This segment dips down into the membrane, forms a tight little loop, which we call a P-loop for pore loop, and then comes back out.
Okay, I'm with you.
When the protein folds into its final 3D shape, the 4 P-loops from the 4 domains cluster tightly together in the exact center to form the actual core. This is the physical, microscopic tunnel that the sodium ions pass through.
And earlier you mentioned that they don't just open, they actively inactivate. The plug swings shut.
Yes. Between the 3rd and 4th domains on the inside of the cell, there is a short intracellular loop of amino acids. This loop acts like a hinged lid. During sustained depolarization, it rapidly swings up and physically occludes the pore, causing that absolute refractory period.
Now, here is where this deep structural biology becomes absolutely critical for pharmacology. Many of the most common drugs that target these sodium channels, like local anesthetics such as lidocaine or major anticonvulsants and mood stabilizers like carbamazepine and lamotrigine, they don't just block the channel indiscriminately. The text refers to them as use-dependent. What does that actually mean?
It is arguably one of the most elegant mechanisms in pharmacology. Use dependence means that the drug does not bind equally to all states of the channel. It binds very weakly to a channel that is quietly resting and closed. However, it binds incredibly tightly to a channel that is actively open or, crucially, in that inactivated plugged state.
I like to think of a use-dependent drug as a highly selective bouncer at a nightclub.
Let's hear it.
So the bouncer walks around the club, completely ignoring the quiet patrons who are just standing at the bar having a drink. He doesn't bother them at all. But the moment a patron starts dancing way too aggressively, throwing elbows, causing a scene, the bouncer steps in, grabs them, and kicks them out.
That analogy captures the clinical brilliance perfectly. If you administered a drug that firmly blocked all sodium channels indiscriminately, regardless of whether they were resting or active, you would immediately stop all electrical activity in the patient's brain and heart. They would instantly go into a coma and suffer cardiac arrest.
Because you'd be shutting down the entire electrical grid.
Exactly. But because drugs like lamotrigine are use-dependent, they spare normal baseline neural activity. When a patient takes lamotrigine for bipolar disorder or epilepsy, their normal cognition, their breathing, their baseline motor functions are largely preserved because those channels spend most of their time in the resting state and the drug ignores them. But when a group of neurons begins firing hyper-excitedly pathological firing, like the runaway high-frequency bursting of an epileptic seizure or the dysregulated hyperactive circuitry driving a manic episode, those specific channels are forced to spend vastly more time in the open and inactivated states.
And that's when the bouncer steps in.
The drug recognizes this aggressive dancing, binds tightly to those specific overworked channels, and clamps down on them. It stabilizes the manic or seizing network without shutting down the entire brain.
That is just pharmaceutical elegance right there.
Yeah.
Okay, so those are the sodium channels, the binary starters. But what about the potassium channels? We mentioned them briefly as the ones that reset the voltage, but the textbook makes it seem like they are the real computational powerhouses.
Potassium channels are the most diverse family of ion channels by far. There are at least 75 different specific types of potassium channels expressed in various cells across the human body. If sodium channels start the fire, potassium channels are the ones that shape the fire, control its duration, dictate how fast it spreads, and eventually put it out.
Let's run through a few of the major functional families here, instead of just a list, because their evolutionary purposes are fascinating. We already mentioned the delayed rectifiers, which pull the voltage down after a standard action potential. But the text says there are specific versions of these that determine maximum firing frequency.
Yes. For example, in the cerebral cortex, we have specific inhibitory cells called fast-spiking interneurons. They are equipped with a highly specialized subtype of delayed rectifier potassium channel that opens and closes incredibly fast. We are talking fractions of a millisecond.
Wow, that is fast.
This allows the cell to repolarize instantly, escaping the refractory period immediately, so it can fire again just a millisecond later. This is what allows for the very high-frequency gamma signaling we see in active cognition.
Then there are the calcium-activated potassium channels, the BK and SK types. These blow my mind because they aren't just listening to voltage, they are listening to the chemical environment inside the cell. They are dual sensor channels.
They represent a beautiful, self-regulating feedback loop. When a neuron fires rapidly, repeatedly over a short period, calcium ions inevitably build up inside the cell with each action potential. That intracellular calcium physically binds to these BK and SK potassium channels from the inside, forcing them open. As they open, potassium rushes out, deeply hyperpolarizing the cell and essentially slamming on the brakes.
So the harder the cell works, the harder these channels try to shut it down.
Exactly. This phenomenon is called accommodation, or spike frequency adaptation. It is why a neuron, when exposed to a continuous, sustained stimulus, will fire rapidly at first, but then progressively slow down and become sluggish.
It's a safety mechanism.
It's a built-in biological governor to prevent overexcitability and excitotoxicity. And interestingly, from a psychiatric perspective, genetic variants in SK channels have been tentatively linked to susceptibility in schizophrenia, and defects in the larger BK channels are associated with certain forms of autism and intellectual disability. When the safety brakes are genetically faulty, network processing becomes chaotic.
That's incredible. What about M channels? The text highlights them specifically.
M channels are slowly activated potassium channels, but their defining feature is that they're actively inhibited by the neurotransmitter acetylcholine.
Right, acting through muscarinic receptors, which is where the letter M comes from.
Correct. When active, M channels act as a slow, continuous brake on repetitive firing. But when acetylcholine binds to the neuron, it closes these M channels, effectively removing the brake. The neuron suddenly becomes much more excitable and responsive.
Like taking the parking brake off a car that's already idling.
Yes, and we know exactly what happens when these channels are structurally defective. Mutations in the genes encoding M channels, specifically the KCNQ subunits, cause a devastating disorder called benign familial neonatal convulsions, or BFNC.
Oh, that sounds awful.
It is a rare genetic epilepsy that presents in newborns within days of birth. But understanding this exact genetic channelopathy has directly driven pharmacology. We now have an anticonvulsant drug called retigabine that works precisely by forcing these defective M channels open, artificially enhancing the brake and stopping the seizures.
I love when the molecular mechanism draws a straight line to the clinical treatment. I have to ask about KER channels, the inward rectifiers. Because unlike all the others we've discussed that let potassium out to hyperpolarize the cell, these seem to do the exact opposite. And the text notes they are heavily used by astrocytes, the support cells of the brain, not just neurons.
This is a vital piece of neurobiology. Astrocytes are non-neuronal glial cells that wrap around synapses and blood vessels. They are packed with inward rectifier potassium channels, or KER channels.
Why does a support cell need a specialized potassium channel?
Think about the metropolis during rush hour. When thousands of neurons in a local circuit are firing wildly, they are constantly repolarizing by dumping massive amounts of potassium out into the tiny, microscopic, extracellular space between the cells.
And if that potassium just sits there and builds up?
It would be disastrous. If extracellular potassium levels rise too high, it alters the chemical gradient for every single neuron in the vicinity. The neurons wouldn't be able to repolarize. The entire local network would become pathologically depolarized and erupt into a massive seizure.
So what do the astrocytes do?
Astrocytes use their KER channels to prevent this. They act like biological vacuum cleaners. They open their KER channels, siphon that toxic excess potassium away from the hyperactive synapses, draw it into their own massive cellular network, and distribute it safely away to areas of lower activity or dump it into the bloodstream. It's called potassium spatial buffering, and without it, our brains would constantly be seizing.
Okay, so we've covered the binary starters, the sodium channels. We've covered the diverse modulators and safety brakes, the potassium channels. That brings us to the third major gatekeeper, calcium.
Calcium holds a unique and profoundly powerful role in the neuron. Sodium and potassium are primarily used just to change the electrical voltage. They are transient electrical signals. Calcium, however, is both an electrical signal and a profound, long-lasting chemical messenger.
Right.
When calcium enters a cell, it acts as a secondary messenger. It alters the activity of enzymes, it physically turns genes on and off in the nucleus, and most importantly for our narrative, it is the absolute trigger for chemical neurotransmitter release.
The textbook divides calcium channels into two broad functional camps, low voltage activated, or LVA, and high voltage activated, HVA. Let's start with the low voltage ones.
LVA channels are also widely known as T-type calcium channels, with the T's standing for transient. As their name suggests, they open in response to very, very small depolarizations. They open at voltages well below the normal threshold required to trigger a massive sodium action potential.
So they're opening when the cell is basically just resting, or barely being nudged.
Exactly. Because they open near the resting state, they aren't used for rapid communication. Instead, they are fundamental for creating intrinsic, rhythmic, pacemaker activity in the brain. They drive the slow, pulsing, oscillating rhythms that control sleep-wake cycles and baseline cortical arousal.
We will come back to exactly how they act as pacemakers in a bit, because the mechanism is wild. But what about the high-voltage activated ones, the L, N, PQ, and R types?
These HVA channels are the heavy lifters of synaptic communication. They require a massive, strong depolarization to open, like the arrival of a full-blown sodium action potential at the end of the axon. These are the channels located predominantly at the presynaptic terminal.
Okay.
When the action potential arrives like a crashing wave, these HVA channels fly open. Calcium floods into the nerve terminal, and that influx of calcium is the physical event that translates the electrical action potential into the release of a chemical neurotransmitter into the synapse.
Which is the perfect transition point in our story. The electrical train has sprinted down the axon, skipping along the myelin subway, and it has hit the physical end of the line. But there is a gap, a physical void between this neuron and the next one. Electricity cannot just jump across empty space in a biological system. So how does the signal cross the void? We move now to section 4, the transfer stations. Synapses, receptors, and chemical signaling.
The influx of calcium through those HVA channels is the absolute non-negotiable requirement for chemical transmission. Inside the terminal, neurotransmitters are packaged into tiny spherical balloons called vesicles.
Right.
When calcium floods in, it binds to specialized sensor proteins, which act like mechanical winches. They physically drag the vesicles to the cell membrane, force them to fuse, and violently dump their chemical cargo, the neurotransmitters, into the synaptic cleft.
The cleft being the microscopic ocean of fluid between the two cells.
Yes. The neurotransmitter molecules float across the cleft and bind to receptors on the membrane of the receiving cell, the postsynaptic neuron. And once they bind, we see two fundamentally different speeds of transmission, fast and slow.
Let's talk about fast transmission first. This relies on ligand-gated ion channels. We talked about voltage-gated channels earlier, where electricity is the key. Here, the chemical neurotransmitter is the physical key, and the receptor is a locked door. The key floats over, slides into the lock, the door instantly flies open, and ions rush through.
Exactly. It is a direct mechanical process. The receptor is the ion channel. The entire process, from transmitter release to ion flow in the next cell, takes only a few milliseconds. It is the basis for all rapid sensory perception, motor commands, and active thought.
And then you have slow transmission, which relies on G-protein-coupled receptors, or GPCRs.
With slow transmission, the receptor is not an ion channel. When a neurotransmitter like serotonin or dopamine binds to a GPCR, it doesn't open a door. Instead, it triggers a cascade of chemical reactions inside the receiving cell.
A cascade.
It activates a G protein, which then wanders off to activate secondary messengers, which might eventually open an ion channel elsewhere, or might travel to the nucleus to change which proteins the cell is manufacturing. This process is slow. It takes seconds, minutes, or even hours to manifest. It is about modulating the long-term state of the cell, governing mood, arousal, and gradual learning, rather than rapid thought.
Let's stick with the incredibly fast ligand-gated channels for a moment. Historically, we classify them by what they do to the receiving cell. We talk about excitatory transmitters like glutamate and acetylcholine, which usually open non-selective cation channels.
Right. When glutamate binds, the channel opens and lets a mix of positively charged ions, mostly toluene, flow rapidly into the receiving cell. This sudden influx of positive charge depolarizes the receiving cell, pushing its voltage up towards that critical minus 45 millivolt threshold, urging it to fire its own action potential.
And on the flip side, we have inhibitory transmitters, primarily GABA in the brain and glycine in the spinal cord. When they bind to their fast receptors, they do the exact opposite. They open chloride channels.
Because chloride is a negatively charged ion, and under normal mature conditions, its concentration is higher outside the cell, opening those doors allows negative charge to flow into the neuron. This deeply hyperpolarizes the cell, pulling its voltage far away from the firing threshold. It acts as a massive wet blanket, shunting and silencing any incoming excitatory signals.
I really want to spend a moment on the structural biology of these receptors, because when you read the textbook, the molecular architecture isn't just dry chemistry, it's practically alien machinery. Let's contrast two major families, the cis loop pentamers and the ionotropic glutamate receptors.
The cis-loop family includes the classic inhibitory GABA receptors, as well as glycine and fast nicotinic acetylcholine receptors. They are referred to as pentamers because they are constructed from 5 separate massive protein subunits arranged in a perfect circle, much like the wooden staves of a barrel, forming a central pore running down the middle.
And here's the question that always baffled me.
Yeah.
How does a tiny microscopic pore know which ion to let through? How does the acetylcholine version of this barrel know to let positive sodium in, while the identical-looking GABA barrel knows to only let negative chloride in?
It's remarkably simple, yet profoundly elegant. It is entirely determined by the specific amino acids lining the inside walls of that central pore. In a GABA receptor, about halfway down the barrel, there is a tight ring of positively charged amino acids facing inward.
And it gives opposite sex.
Exactly. When the channel opens, that positive ring acts like a magnetic sorting tube. It aggressively draws the negatively charged chloride ions through the pore, while violently repelling any positively charged sodium or potassium ions that try to enter. In an acetylcholine receptor, that ring is negatively charged, so it pulls sodium through and repels chloride. The receptor doesn't know anything, it just applies basic electrostatic physics.
That is brilliant. But the glutamate receptors, the main excitatory workhorses of the brain, are built completely differently.
Yes. The AMPA and NMDA receptors, which mediate almost all fast excitatory transmission in the cortex, are not pentamers. They are tetramers built from 4 massive subunits. And advanced crystallographic studies show they have a truly bizarre, fascinating shape extending far out into the extracellular space.
The text describes the AMPA receptor as looking somewhat like the letter Y, and the actual binding site where the glutamate attaches looks like a Venus flytrap.
Or perhaps a clamshell is a better mechanical analogy. The binding site consists of 2 lobes separated by a deep cleft. The glutamate molecule floats in and binds deep within that cleft.
And then the clamshell physically snaps shut around it.
It snaps shut. And because the bottom half of the clamshell is tethered directly to the membrane-spanning segments of the protein, the physical snapping shut of the cleft generates a mechanical torque. It literally wrenches the transmembrane segments apart, forcing the ion pore open. It is a direct translation of chemical binding into mechanical leverage.
I have a massive pushback here, though. Something that completely upends how we usually talk about this.
Yeah.
In psychiatry, and in basically every introductory neuroscience class, we constantly refer to glutamate as excitatory and GABA as inhibitory. It's treated as a fundamental law.
Right.
But wait, is the neurotransmitter molecule itself inherently excitatory or inhibitory?
That is a brilliant question to raise, and the answer is a resounding categorical no. A molecule of GABA is just a small string of carbon, hydrogen, nitrogen, and oxygen. It has no intrinsic inhibitory magic. The neurotransmitter is simply a physical key. The resulting action, whether the cell is excited or inhibited, depends entirely on the architecture of the receptor it binds to and, critically, upon the ion gradients of the receiving cell.
This brings us to what I consider the most mind-bending, counterintuitive fact in the entire source chapter. GABA is known universally as the brain's main inhibitory neurotransmitter, but the textbook explicitly notes that early in fetal development, in the growing brain, GABA is actually excitatory. It causes action potentials. How is that physically possible?
It represents a profound paradigm shift in how we understand neurobiology, and it all traces straight back to the fundamental physics we discussed in Section 1, chemical gradients. In a mature adult neuron, the cell actively pumps chloride ions out into the extracellular space, so when GABA opens a chloride channel, chloride rushes in, carrying its negative charge, and hyperpolarizes the cell. That is inhibition.
Okay, but in a fetus?
In a developing immature fetal neuron, the cell expresses an entirely different set of ion transporters. Instead of pumping chloride out, it actively pumps chloride into the cell. So the intracellular concentration of chloride is massive.
Oh, wow. So when a molecule of GABA floats over and opens the channel in a fetal brain?
The massive chemical gradient forces the negatively charged chloride to rush over the cell. The cell is rapidly losing negative charge, which means the inside of the cell is becoming significantly more positive. It depolarizes, it fires. In the developing brain, GABA acts as a crucial, widespread excitatory signal that drives the spontaneous electrical activity necessary to wire up the newly forming cortical circuits. Only as the brain matures do the genes for the mature chloride transporters turn on, the chloride gradient reverses, and GABA flips to becoming inhibitory.
But the clinical implications of this are terrifying. The expert text notes that mature neurons aren't permanently locked into this state. Severe physical stress or pathologies like brain tumors can actually cause mature neurons to revert to their immature fetal state. They change their transporter expression.
Yes. Under severe pathological stress, like the physical trauma of a severe traumatic brain injury, the hypoxia of a stroke, or the metabolic disruption of a growing glioma, the stressed neurons can lose their ability to pump chloride out. Intracellular chloride levels slowly begin to rise.
And then, when the surrounding network tries to calm the stressed area down by releasing GABA,
The GABA opens the channels, but because the chloride gradient is broken, chloride flows out instead of in. Suddenly, the GABAergic interneurons that are desperately trying to calm the network down and prevent a seizure are actually releasing GABA that causes massive excitation.
it's like pressing the brake pedal in your car as hard as you can and discovering the mechanic wired it to the accelerator.
Exactly. This trauma-induced reversal of the chloride gradient is currently thought to be a major fundamental driver of epileptogenesis, the progressive creation of a seizure disorder, in patients following severe head injuries or in the tissue surrounding brain tumors. It fundamentally changes how we view the pharmacology of treating these specific types of seizures.
Okay, let's take a breath and zoom out. We have meticulously built the individual components of our metropolis. We understand the energetic tension of the resting battery. We understand the high-speed trains of the action potentials. We have mapped out the incredibly diverse gates of the potassium and sodium channels. And we understand the mechanical clamshells and sorting tubes of the chemical transfer stations. We're moving now to Section 5. Systems level. Excitation-inhibition balance, network oscillations, and the catastrophic breakdowns of channelopathies. What actually happens when you wire a billion of these components together into a city grid?
When you network billions of these cells together, the result is not just chaotic noise. You get emergent, highly synchronized network rhythms, oscillations, brain waves. And these global rhythms are largely dictated by specific microscopic pacemakers embedded within the circuits.
The textbook uses the inferior olivary neurons in the cerebellum as the classic example of a pacemaker. These are the cells that regulate our fine motor timing and learning. The text gives a beautiful sequential breakdown of how they generate a continuous, ticking biological clock.
It is a self-sustaining, continuous loop of ion channel handoffs. First, the cell fires a standard high-voltage sodium action potential. That massive depolarization forces open the high-voltage activated calcium channels. Calcium rushes in.
Okay, spike in calcium influx.
Now remember those calcium-activated potassium channels we discussed earlier, the safety brakes. The sudden flood of intracellular calcium binds to them, forcing them open. They cause a massive efflux of potassium, which severely and deeply hyperpolarizes the cell, driving the voltage way down.
So the cell fires a massive spike and then immediately and aggressively shuts itself down into a deep freeze.
Yes, but here is the final brilliant twist. Do you remember the other calcium channels we discussed? The low-voltage activated T-type calcium channels?
The ones that operate near the resting state.
Exactly. But T-type channels have a unique quirk. They are completely inactive at normal resting voltages. They only become primed, unlocked, and ready to open when the cell is subjected to a deep, prolonged hyperpolarization. So the massive hyperpolarization caused by the potassium channels physically primes the T-type channels.
Oh. So as the potassium channels slowly close and the cell's voltage naturally begins to drift back up from the deep freeze.
As it drifts up, it crosses the very low threshold for the now primed T-type channels. They snap open. A slow trickle of calcium leaks in, gently depolarizing the cell until it hits the main threshold, and boom, it triggers the next massive sodium spike. And the cycle repeats endlessly.
Sodium spike, massive calcium influx, calcium-triggered potassium deep freeze, which primes the T-type calcium channels, which slowly depolarize the cell to trigger the next sodium spike. It is a perfectly engineered biological clock ticking away in the cerebellum.
And we see the exact same logic elsewhere. In the thalamus, massive networks of similar T-type calcium channels drive the slow, synchronized rhythmic brainwaves that govern deep restorative sleep. But if those thalamic T-type channels suffer a genetic mutation or become pathologically dysregulated, they generate an abnormal, hypersynchronous 3 Hz spike and wave pattern across the entire cortex.
Which is the exact EEG signature of childhood absence epilepsy,
Yeah.
where a child just suddenly stops and stares blankly for 10 seconds as the pacemaker hijacks their consciousness.
Precisely, which brings us to a major unifying framework in modern psychiatric research, the excitation inhibition, or EI balance hypothesis.
The premise here, as I understand it, is that complex, healthy behavioral states, sustaining attention, regulating mood, navigating complex social interactions, require a perfect, delicate, microscopic balance between the excitatory glutamatergic signals pushing the network forward and the inhibitory GABAergic signals holding it back.
It is a razor's edge. And in complex psychiatric disorders, particularly schizophrenia and severe autism spectrum disorders, there's growing, robust evidence that this critical EI balance shifts pathologically toward overexcitation, particularly within the highly evolved circuits of the frontal cortex and hippocampus.
But the textbook makes a crucial distinction. It's not just a general loss of all GABA. The key players maintaining this specific balance are a very specialized subpopulation of inhibitory cells, the parvalbumin, or PV, positive interneurons.
PV interneurons are the elite conductors of the cortical orchestra. They are fast-spiking cells. They physically wrap their synaptic terminals directly around the cell bodies of hundreds of excitatory pyramidal neurons, exerting immense control. And because they are equipped with those extremely fast-delayed rectifier potassium channels we mentioned, they can fire at incredibly high frequencies without fatiguing.
They are the ones responsible for generating gamma oscillations, right?
Yes. Gamma oscillations are high-frequency brainwaves, usually between 30 and 80 hertz. When you are intensely focusing on a complex task, actively processing sensory information, or locking in a memory, your cortical networks synchronize into a gamma rhythm orchestrated entirely by the rapid-fire pacing of the PV interneurons.
So if your PV interneurons are genetically faulty or damaged by inflammation, you lose that tight gamma synchrony. The network gets noisy, uncoordinated, and chaotic. Information transfer breaks down, which manifests clinically as the severe cognitive deficits, the working memory failures, and sensory processing issues we see in patients with schizophrenia.
Exactly. The hallucination isn't just a psychological metaphor. It is the result of a noisy, desynchronized sensory cortex. And we have fascinating pharmacological models that recreate this exact state. Let's look at ketamine.
Ketamine is everywhere in psychiatry right now as a rapid antidepressant, but traditionally it's a dissociative anesthetic. At subanesthetic doses, it induces profound dissociation, a feeling of detachment from reality, and sometimes overt psychotic-like symptoms. How does it do that?
For decades, we simply said it blocks NMDA receptors. But recent high-resolution studies show a much more nuanced picture. Ketamine strongly targets specific pacemaking channels, including those HCN1 channels we briefly touched on, the hyperpolarization-activated channels that help set baseline rhythm.
It blocks them.
It does. And in very specific, highly connected areas of the brain, like the retrosplenial cortex, which acts as a major hub for maintaining vigilance and integrating your conscious sense of space and reality, blocking these HCN1 channels induces a profound, abnormal, slow 1 to 3 hertz rhythm.
The circuit essentially goes offline and starts humming to its own pathological disconnected tune.
Yes. The retrosplenial cortex literally disconnects its rhythm from the rest of the brain. And what is truly astounding is that a team of researchers recently recorded an almost identical, highly localized 3 hertz rhythm in the deep brain structures of a human epilepsy patient in the exact moments they were experiencing a pre-seizure aura characterized by intense dissociation and out-of-body experiences. It perfectly correlates a cellular ion channel mechanism to one of the most complex, difficult-to-describe psychiatric symptoms imaginable.
This concept linking complex systemic illnesses directly to the failure of specific ion channels is called a channelopathy, and the source material highlights how psychiatry and neurology are increasingly viewing a massive range of disorders entirely through this microscopic lens.
We mentioned the CACNA1A gene earlier in passing. It encodes the main pore-forming subunit of the PQ-type high-voltage calcium channel, the very channel responsible for releasing neurotransmitters at the synapse. Mutations in this single specific gene can cause a staggering, almost unbelievable array of severe neurological issues.
The textbook lists familial hemiplegic migraine, where patients get severe migraines accompanied by sudden, stroke-like paralysis on one side of their body.
Yes, and completely different mutations in the exact same gene cause episodic ataxia, a profound temporary loss of all motor coordination, or progressive cerebellar degeneration. It vividly demonstrates how absolutely critical precise calcium entry is at the synapse. Alter the pore slightly, and the entire motor or vascular system collapses.
What about the pain pathways? The text tells an incredible story about the SCN9A gene.
SCN9A codes for a specific type of sodium channel known as NaV1's 1.7. This channel is heavily, almost exclusively, expressed in the peripheral sensory neurons that detect tissue damage and transmit pain signals to the spine.
So if you have a genetic mutation that causes this NaV1.7 channel to inactivate too slowly, meaning the plug doesn't swing shut fast enough, and the channel stays open and fires far too easily.
The patient suffers from primary erythromelalgia or paroxysmal extreme pain disorder. Their pain-sensing neurons are hyperexcitable. A slight change in temperature or a light touch can trigger agony. But conversely, if a patient inherits a mutation that completely breaks the channel, a total loss of function where the Nav1.7 channel cannot open at all, you get a condition called congenital insensitivity to pain.
People with this mutation can literally break bones, bite through their own tongues, or suffer severe burns and feel absolutely nothing.
Complete analgesia simply because the sodium channel required to send the signal to the brain is physically broken.
And these channelopathies aren't just inherited genetic destiny, are they? They can be acquired later in life, often through catastrophic autoimmune errors.
This represents one of the most important paradigm-shifting developments in biological psychiatry over the last 20 years, anti-NMDA receptor encephalitis.
Walk me through what this actually looks like in a clinical setting.
Typically, a patient, often a young woman, sometimes with an underlying ovarian teratoma, but often with no identifiable trigger, will present to the emergency department with what looks exactly like a rapid-onset, severe, acute psychotic break. They suffer from intense paranoia, bizarre delusions, auditory and visual hallucinations, and severe behavioral agitation.
For decades, these patients would have been immediately locked in a psychiatric ward and diagnosed with a primary catastrophic onset of schizophrenia.
Exactly. And the antipsychotics wouldn't work. As the days progress, the psychosis devolves into a severe delirium, profound movement disorders, dysautonomia, where their heart rate and blood pressure swing wildly, and eventually, intractable seizures and coma.
Because it isn't primary schizophrenia, it is an autoimmune channelopathy.
Yes. The patient's immune system has mistakenly produced antibodies that specifically target and attack their own NMDA glutamate receptors in the brain.
How do the antibodies actually stop the receptors from working? Do they block the pore?
It's more destructive than that. The antibodies bind to the NMDA receptors on the surface of the neuron and cross-link them together. The neuron recognizes this massive abnormal protein clump on its surface and internalizes it. It physically pulls the NMDA receptors inside the cell and destroys them, effectively sweeping them completely out of the synapse.
And NMDA receptors, as we know, are the fundamental excitatory workhorses crucial for network stability, learning, and reality testing.
Without them, the entire cortical network destabilizes and collapses into noise. But the miracle of this discovery is that if the psychiatrist recognizes the specific clinical progression, tests the spinal fluid for the antibodies, and treats the patient with aggressive immunotherapies, steroids, plasma exchange, drugs that wipe out the antibody-producing B cells, the brain eventually manufactures new NMDA receptors, repopulates the synapses, and the patient can make a complete, seemingly miraculous recovery from profound psychosis and coma back to normal function.
Which is an incredible, hopeful segue into our final major section. Section 6. Active interventions. Plasticity and brain stimulation. If the city grid is malfunctioning, whether from inherited genetics, physical trauma, or an autoimmune attack, how do psychiatrists try to physically repair it? We transition from understanding the pathology to mastering the mechanisms of therapeutic intervention, and that requires a deep dive into plasticity.
For a long time, the brain was viewed as a hardwired static machine. But the reality is that it is constantly, continuously remodeling itself based on experience. When we talk about how the brain learns, how it forms a memory, or how psychotherapy might alter a fear response, we usually talk about Hebbian plasticity.
Named after the psychologist Donald Hebb, and usually summarized by the famous phrase, cells that fire together wire together.
Yes. This includes the biological processes of long-term potentiation, or LTP, where a synapse becomes permanently stronger, and long-term depression, or LTD, where it weakens. Hebbian plasticity requires the precise, simultaneous firing of both the presynaptic neuron sending the signal and the postsynaptic neuron receiving it. It is almost always mediated by a massive influx of calcium through those NMDA receptors, which then act as biological coincidence detectors, turning on genes to build more receptors and physically enlarge the synapse.
But the textbook emphasizes a huge logical flaw if the brain only used Hebbian plasticity.
The flaw is runaway excitation. If synapses just keep getting stronger and stronger every single time you learn something or experience something, eventually the dominant synapses would become so massive and excitable that the entire network would lock up into a continuous, massive seizure. The brain would explode from its own learned connections.
Which introduces the necessity of homeostatic plasticity.
If Hebbian plasticity is about changing specific individual synapses to store a discrete piece of information, Homeostatic plasticity, also called synaptic scaling, is the brain's global mechanism for maintaining the overall EI balance. It operates slowly, over hours or days. If a neuron has been overly excited for too long due to LTP, the cell will actively scale down the strength of all its excitatory synapses proportionally and perhaps increase its inhibitory GABA receptors. It constantly adjusts the baseline to keep the neuron's average firing rate within a safe, optimal, and biologically sustainable range.
So Hebbian is about learning the facts, and homeostatic is about keeping the overall system stable so you don't burn out.
Yeah.
And then the text mentions metaplasticity, which is just a wonderful sci-fi sounding term. The plasticity of plasticity.
It is the brain actively adjusting how easy or how hard it is to induce Hebbian learning in the first place. For instance, chronic severe stress alters a neuron's metaplasticity, raising the threshold, making it much, much harder to induce LTP and learn new adaptive behaviors, while making it easier to induce LTD and weaken connections. This explains why patients in deep depression find it physically difficult to learn, adapt, or change their thought patterns. The machinery of learning has been downregulated.
Interestingly, the textbook notes that our most rapid-acting psychiatric treatments like the ketamine we discussed, or classic mood stabilizers like lithium, seem to operate heavily via these non-Hebbian homeostatic mechanisms. They aren't erasing specific traumatic memories. They are forcefully resetting the network's overall baseline excitability.
They'll restore the capacity for plasticity, allowing the brain to heal itself.
Speaking of forcefully resetting the network, let's talk about the ultimate neuromodulation, electroconvulsive therapy, or ECT. It is a treatment historically fraught with stigma in the public imagination, often viewed as barbaric. But when you look at the biophysics described in this chapter, it is not just indiscriminately shocking the brain.
Not at all. Modern ECT is highly precise, and the actual physics of the electrical wave matter immensely for patient safety. The clinical goal of ECT is to deliberately induce a generalized synchronous seizure in the brain, because we know that the intense physiological process of the seizure itself is what triggers a profound therapeutic cascade. It releases massive amounts of brain-derived neurotrophic factor, forces massive neurogenesis in the hippocampus, and violently resets the homeostatic plasticity of the cortex.
But the clinical challenge is, you want to trigger that seizure using the absolute minimum amount of external electrical energy possible to avoid causing collateral tissue damage or severe memory loss.
Exactly. And this is where the history of the electrical waveform is so illuminating. In the early historical days of ECT, the machines delivered sine waves. A sine wave is a smooth, continuous, undulating wave of alternating current, exactly like what comes out of a standard wall socket in your house.
Why is a sine wave bad for stimulating the brain?
It is highly dangerously inefficient. First, the voltage of a sine wave rises slowly, like a gentle hill. This slow rise gives the neuron's membrane time to actively accommodate to the change, meaning you have to use a much higher peak voltage to finally force it to fire. But the real problem is what happens next.
The refractory periods.
Yes. The sine wave continues continuously pouring electrical energy into the brain tissue for several milliseconds, long after the neurons have already fired their initial action potential. It is pumping electricity into the cells during their absolute and relative refractory periods when their sodium channels are inactivated and physically locked shut.
So pouring electricity into a neuron during its refractory period does absolutely nothing to help trigger the seizure.
It accomplishes nothing therapeutically. It simply heats up the brain tissue, causes neuroinflammation, and drives the severe, long-lasting cognitive and memory side effects that plagued early ECT.
So what is the modern biophysical solution?
Brief square wave pulses. Instead of a continuous rolling wave, modern ECT machines deliver a very sharp, sudden, vertical spike of voltage that holds flat for a tiny fraction of a millisecond, usually 0.1 to 2 milliseconds, and then drops instantly back to zero. The machine waits and then delivers the next pulse.
It's like a strobe light instead of a continuous floodlight.
Precisely. The sharp vertical rise hits the neurons instantly, bringing them to threshold before they can accommodate. And more importantly, the electricity turns completely off while the neurons go through their refractory period. The machine gets out of the way, waits for the cells to reset, and then hits them again. It is about actively entraining the neurons by mimicking their own natural, discrete firing patterns, rather than just overwhelming them with raw power.
The text mentions that for unilateral ECT, where you only stimulate the non-dominant hemisphere of the brain to preserve verbal memory, they now use ultra-brief square pulses, lasting around 0.2 milliseconds. But you have to dose them massively high, at 5 to 6 times the patient's individual baseline seizure threshold to ensure efficacy.
Yes. It highlights a critical lesson. In neuromodulation, the electrical dosage, the physical shape, duration, and frequency of the wave is just as deeply critical as the chemical dosage in milligrams of a pill. And this logic extends perfectly to deep brain stimulation, or DBS, which we use for Parkinson's, and increasingly for severe OCD or depression.
DBS involves surgically implanting electrodes deep into specific brain nuclei.
And we don't use sine waves there either. DBS uses continuous high-frequency square wave pulses, often over 100 hertz. For a long time, we thought DBS simply turned off the hyperactive brain area by jamming it. But advanced computational modeling suggests something much more elegant. The high-frequency stimulation suppresses the chaotic, intrinsic firing of the cell bodies, but it actually entrains and enhances the regular firing of the axonal outputs. It functionally overrides the pathological, noisy rhythms of the disease circuit with a clean, high-frequency artificial hum.
We have covered an absolutely massive amount of ground today. We've journeyed from the incredible energetic tension and metabolic cost of the resting potential through the explosive speed of the insulated action potential. We've navigated the incredibly diverse gatekeeping of the sodium, potassium, and calcium channels and decoded the mechanical architecture of the synapses. And we've seen how these microscopic elements scale up into the network-level rhythms that dictate our consciousness, our sleep, and the catastrophic breakdowns we see in clinical channelopathies.
To really cement these concepts, because knowing the precise mechanism is the absolute foundation of your clinical reasoning moving forward, I think it's vital we quickly synthesize the key takeaways. Let's make sure the core concepts are locked in. I'm going to pose a few questions to you listening and give you a brief moment to think. First, what accounts for the massive energy consumption, up to 40% of brain oxygen at the resting membrane potential?
Think about the boat analogy.
The answer is the sodium-potassium AT-pace electrogenic pump constantly working against the gradients to maintain cellular homeostasis. Next question. Why do local anesthetics and certain anticonvulsants selectively target hyperactive neurons while sparing normal resting activity?
The bouncer at the club.
Right. It's the use-dependent blockade of sodium channels. The drugs bind tightly to the open or inactivated states of the channel. Question 3. How can a GABAergic input theoretically trigger an excitatory response, as seen in early development or certain stress pathologies?
This one is wild.
It's due to the reversal of the intracellular chloride gradient. High intracellular chloride means GABA, opening the channel, causes chloride to exit, depolarizing the cell. Okay, question 4. Which type of calcium channel is most directly responsible for the low-frequency oscillatory pacemaker rhythms seen in the thalamus during sleep and absence epilepsy?
The ones that need a deep freeze to prime.
The LVA, or T-type calcium channels. Question 5. When administering unilateral ECT, why is a long sine wave stimulus less effective and more prone to side effects than an ultra-brief square wave pulse?
Strobe light versus floodlight.
Sine waves deliver current during absolute and relative refractory periods, failing to efficiently entrain neuronal firing while causing excess thermal damage. Final question. In the EI balance hypothesis, which specific interneuron type is critical for generating the high-frequency gamma oscillations thought to be disrupted in schizophrenia?
The conductors of the orchestra.
The parvalbumin-positive fast-spiking interneurons. I strongly direct you back to the Kaplan and Sadok chapter to review the nuances of these mechanisms.
I completely agree. The visual models make the abstract chemistry tangible. I want to leave you with a final thought to ponder as we wrap up this deep dive. If our entire subjective reality, our deepest memories, our most profound traumas, our capacity for joy, our very consciousness is ultimately dictated by the microscopic opening and closing of protein gates in response to the flow of simple charged ions, at what point does chemistry become mind? We are rapidly entering an era where techniques like optogenetics and chemogenetics are allowing scientists to control these specific channels in living animal models using flashes of targeted laser light or designer drugs. They can physically turn specific memories or complex behaviors on and off like a light switch by targeting single-ion pores. How long until we can precision engineer human emotion at that level? It's a thrilling and frankly terrifying frontier.
It is the ultimate frontier of medicine, because psychiatry at its absolute core is the study of how the physical brain, the subjective mind, and the lived experience interact, and how that profound, delicate process can go wrong.
Here are the details of this episode.
Cellular and synaptic basis of neural signaling. Byline. The language of the brain, how cells communicate, and meaning begins to emerge.